as kind of a proof of principle, they showed that they had one of their models. They showed it the Wikipedia entries for different genes, and then asked it to go ahead and create a Wikipedia entry for every gene. And that's available on wikicrow.ai.
Oh, cool.
And they had an independent panel of scientists compare or rate the Wikipedia entries with the WikiCrow entries. And
Oh, that's a big, wow. Yeah.
And the WikiCrow entries were judged superior in
Interesting.
most dimensions. So more complete, more authoritative, easier to read. So.
Well, that's quite serendipitous that you mentioned that, because I think that will probably factor into our conversation later today.
Excellent.
As part of our discussion. All right. 10:01 or 50, no, time is it 3:01, 3 o'clock. All right. I see lots of faces. I think we're gonna go ahead and get started. Yeah, I think we have quorum. You know, people are allowed to roll in a little late. That's fine. They're not gonna miss much. The action will really happen later. Okay, I'm gonna share my screen. I'm kicking us off today. Let me see if it works though first. Can everyone hear me on the phone? You can thumbs up or unmute yourself. Yeah, okay, awesome. Because we're in a big room here in Boston. So I want to make sure you can hear me. All right. Fabulous. Everyone see that okay? I'll switch to mode. No. Wonderful. Okay, as people are joining, we can just join and jump on. You'll know exactly what we're talking about right away. I want to thank you all for joining us at our official kickoff to our working group. That was something we said we'd do back in September. We actually did, which is great. The goal of this working group is to think about standards and infrastructure for predicted effector gene lists. And I'm going to give a little intro, and then we're going to dive into the agenda today. But really, most of today's conversation is going to be actual conversation and trying to actually work through this problem and this challenge. First of all, I want to thank the organizers of our first workshop, which all of you probably attended. And it was actually really a fun thing for us to do, and I think for the community. We're going to give you a little recap of that today. And I also want to thank the folks from today who really made today happen. Julie, Aoife, Laura, and Yue, who you're all going to hear from over the course of the day.
دخل اجتماع ساعتين.
What'd you say?
دوي.
I think some people are unmuted. Yeah.
I'll go through and mute.
Yeah, that's okay. No worries. We don't mind. We're a working group, remember. It's a little bit more low key for sure. But we are going to record everything from today and keep notes and things like that. So that's like the basics. And this is not a webinar. This is an active chat. We're in multiple locations. So please be happy to interrupt, raise your hand, whatever method you want to get our attention. We'll be monitoring everything, make sure everyone gets a chance to communicate and share. But, you know, I want to just remind us of what we did back in September. We had a really excellent workshop. I think we sent out a survey and 160 people replied, something like that. When we thought about doing this workshop for community standards for effector gene lists. And when we actually had people attend, about 9 people attended, and it was all over the world. It was bicontinental, it was in person and virtual, which I thought was really excellent. And what I thought was really cool is that, you know, it was very participatory. We got representation from different sectors, folks doing gene lists, folks consuming them, interpreting them, thinking about how to use them in the future. And I love this image that Maria really coined for me. The objectives from that workshop were the following, just to remind you all. I'm sure Aoife's going to go into this, but I wanted to sort of put it out there for us today. You know, just raise awareness about the current landscape back in September. And I think the goal is to really establish a community standard for peg lists. That's what today's about, right? To do that as a community with you all. Ensure that these are fair, accessible to the entire community so they can be useful, right? Define strategies for how you evaluate and collate them as well. And then some of the challenges, right? I think some of the challenges really help us define the standards and benchmarks that we're going to talk about today. And really, finally, strategies to make it super easy for someone to contribute and make their results of these really rich lists acceptable. So the outputs of that meeting I thought were really valuable, really like it was aimed at, you know, talking about a straw man, it was aimed at engaging a community, potentially having a white paper is coming out of it, a resource for both the deposition and the representation, a working group, for making good on that. And ideally a roadmap, because these resources are meant to make it easier for others to use them and make discovery for follow-up, for prioritization, target identification, and so on and so forth. So with that, I think we were successful with our first output. I think we have a better engaged community because we had a lot of people attend, and I can tell you that even better, we have a YouTube channel that we keep where all of our knowledge portals, and we videotaped that workshop, and it's by far the highest views we've ever had. We have 5,800, and as of last month, it was like 5,700. So people are still taking a look at it. I think that's pretty cool. Our average viewers is like 300. So big difference. So thank you. That actually meant something to us, and it also meant something to the community. It's actual evidence that this was valuable to others. So that's pretty cool, and that's available if you ever want to listen to it again. And it's nicely annotated with all the speakers, so if you don't want to listen to me drone on, you can go to Eric, because that's really cool stuff, right? So that's all there in perpetuity. Another one was, resource. So back in September when we did this, we had started, the reason we did this is because at the Knowledge Portal and with our EVI colleagues, we had a couple things happening. GWAS Catalog is the best place in the world to put your summary statistics, right? They were also getting a lot of inquiries from collaborators saying, hey, we have this auxiliary list of prioritized genes and all these evidence sources. Where do we put it? How do we make it useful? We just stick it back in the end of our supplemental figures. As folks at the Knowledge Portal side, we had people saying, I have this really nice visualization I've made with my curated list. How do you make those accessible? So we came together and said, both sides of these coins are very useful to bring together. Let's work on building a resource. However, if you build a resource without folks that know how to do it best, the resource doesn't get used, to find out. So the goal is to really work with you all as community contributors and consumers to ensure that we have the right standards and representation for those. So, we have these lists, they're all accessible. Right now, there's 150 or so on the portal for common metabolic disease and association to function. You can find them here. This gives you an example for like one gene that's like, if you're not only want to search your favorite gene through just the effector gene list, which you have 50 of them, if you're on the portal searching your favorite genes, this is an example for ApoE, you'll see the Hughes scores, right? That's all the genetic evidence. But then below here, you'll see all of the lists that gene appears that we've curated. So this gives you an idea of just where it's acceptable currently. And then you can dive into the actual list itself, and this is always vetted by the contributor. And Maria spent a good portion of the last year of her time at the Broad Institute. She came back out of retirement just to come talk to us today. She curated all these, and it really was that by which we stumbled upon, holy moly, there's a opportunity here. We also, I think we might have forced Maria into retirement trying to make her do all those lists because it was a lot of work, and so many people do it different ways. So we thought that this would be an opportunity for us as centers and with contributors to try and make these standardized and represented in a similar way. So, to that end, we wanted an output that would be useful. Maria's going to talk about some progress we've made on that end in a moment. And also other white papers that want to come out of the output of this group, something written that can be a standard for our community. So our working group today, that's really what we're here to talk about. We're here to shape and share standards with our community. So to do that, we really have to advance these same things, right? We really have to have a manuscript that comes out of this working group's activities. We want to enhance the resource that's sort of already percolating around a place to deposit the evidence sources, the list themselves, the metadata associated with them, and then a resource for representing them in an easy way for someone to interrogate. And ideally, if we do that well, it's a roadmap for discoveries. How do you incorporate all the lists together? How do you mine them? How do you use them yourself? And ideally, make discoveries. So with that, a couple considerations before we dive in, because it's been how many months since we had that? I can't add. Since we had that workshop. So things have changed. And I just thought, what has it been like in the last couple of months? So a couple considerations. I just did a quick, survey of people, what's going on in our community. People are still churning out lists, right? They still are. This is just a sampling of some of the recent ones. These are folks that are doing, diseased focused, curated lists. I actually look for things that were not just informatic method, that were literally a combination of, heuristics, curation, human interaction, and multiple layers of evidence. Evidence sources we're going to talk about later today. But this just gives you an example. There's more for Parkinson's disease and so on and so forth. All over the place. There's new versions of current lists that are coming out there. So the propagation is real. Another thing, you got new approaches, right? These are both curated and computational. Again, this is just a sampling of some you might be familiar with. Another one that came out from Shipper et al. Example is the Flame Score. I like the name, it's fantastic. But again, if you look through all these, they're doing something similar. They're using some of the same methods. They're all trying to get these lists. They're a combination oftentimes of curated and computational, but I'll tell you it's something, no standards, no consistent place where they deposit them. Thanks to Open Targets, for example, they made theirs successful, as you can see here. But that's, again, it's kind of like the Wild West. So this just gives you an example of this, just like what Flames is doing, just to give you a sense. Again, these approaches are complicated, but also not completely novel. They're sort of like different slants and things that have been done, but they are discovering new things that are incredibly valuable to the community. Then another thing that's a really important point, and I love this. This is one of the most beautiful figures I've seen in a long time. It's from Catelyn Schurstak's team. And this is important to me as like a final note. These lists, this list currently exists on the knowledge portal that I mentioned before for all of the curated effector gene lists, but it's an older version. So it's not up to date anymore. She just recently published, the Lou et al. Group published this a couple months ago, and this is more comprehensive, iterative, like more loci, better fine mapping on the loci that they identified. And the only place this is available on Catelyn's website, which happens to be elegant and beautiful. But if you needed this, you'd have to go there, download it, yada, yada. It would be accessible to you, but it wouldn't be something that you can incorporate readily into what you're doing. But it's quite beautiful. So some of the things, I put this up here because some of the visualizations are quite nice. She called it her scorecard, which I really like as well. So the final consideration, upgrades. For those of you who've ever seen The Matrix, this makes sense to you, but in the last year, I don't know if you've noticed, our life has changed when it comes to AI, machine learning techniques, Chat GPT, DTP, did I say it right? I always get thank you. And it's coming into our universe, and to the point where it's like feels like a buzzword. But if we don't embrace this and what we're doing here, I think we would be at a loss. So I want to make sure that's incorporated in our conversation today, particularly because you have not only like a wealth of data that's actually well structured now. That's the key. Like you can go into places, resources like the GWAS catalog, like Open Target, like our portal, where things are curated and handled well. You can put a reasoner on top of that using AI and come out with more discovery, using the reasoner to help you navigate through stuff that's well curated. So we should consider how technology is going to be adopted and make these resources standard for machine learning and AI tools. So finally, let's give this poor man some bones, skin, and a personality today. And I realized that this actually looks like my husband. For those of you who know him. And I think it's because I swear to God, it's because we train. I don't know if we use ChatGPT, we use images all the time, we use the same. Anyways, I thought that was kind of fascinating. Anyways, but with that, here's our agenda today. Is to, we're gonna have a little group exercise at the beginning. We're gonna do a little survey, a poll. Mentee Laura will introduce that for us. We'll do some intros because all of you on the call, this is a working group. We hope we will see each other's faces at an interval over the next, X months while we actually define these standards. Maria is going to give some reflections as a retired geneticist and biocurator from her time here and the work she did here. And then Eva will recap the workshop a little bit more structurally and specifically than I did. Julie will put our charge to the team and what we're going to do today. And then Laura, I think, is going to lead us through the guidelines, sort of starting to get a sense of what the straw man would look like. And then really the important part, group discussion. So that's the landscape. And any questions before we dive in or any programming notes you need to be making? Yeah? Anybody on, the chat's all addressed, I saw some chats.
Yes, so
Awesome.
we have a live meeting notes, so feel free to add anything as we go. I will be taking notes for us as well, but if there's something specific that you want, we will have that available for you. I sent the link in the chat, so if you don't see it, please let me know and I can send it to you.
And we're asking all the participants to please sign in there as well so we can track who is on.
Awesome. All right. With that, will I hand it over to Laura to start off with our first exercise, or is it Julie? Laura? Julie? Laura. All right. I'm going to stop share and turn it over to Laura.
Sure. Okay. So later on in the working group meeting, we will be using Menti to gather feedback from people because we're quite a large group and we want to make sure that everyone has a voice, right? And we want to hear everyone's opinion and gather consensus. So I just want to start and get everyone set up on Menti, I think I need to do it like this. So hopefully you can see at the top of the screen, there is a number. So if you can open a browser or you can use a mobile phone and look at menti.com and then enter the code, which is there on the screen, 37276552. And once you're into the Menti system, please just type in the box to tell us who you are, what your role is, and what institution you're from. And I'm just going to leave that open for a few moments to let people type. So I realize I can't project the Menti and type at the same time. So I'm Laura Harris, and I'm the project lead of the GWAS catalogue at EBI in the UK, in case you don't know who I am.
Those last the 4 digits are 65552.
52. Mhm.
Thank you.
Great. Can see
This
it.
is a neat thing. I like that. It's kind of.
Yeah. Oh, nice.
So I think we've got 21 people in the system now, and we've got
Oh, cool.
responses from almost everyone.
Make the point to Noelle.
So I'm just going to give one more minute. So hopefully that is most people now. So then I'm going to move on to the next slide. But of course, please do add your name to the Google Doc as well at any point during the meeting. But we will be also recording what's put into the Menti as well. So if you make comments here, we'll have that for the record. So on the next slide, I would like to know, what's your relationship to peg lists? So for example, if you're somebody who does your own research and generates peg lists, I would call you a data generator. If you're somebody that uses other people's peg lists, I would call you a data consumer. Or you might be somebody who develops software tools for generating or consuming, analyzing peg lists. I would call you a tools developer. Or maybe you're something else. You might be more than one of these people. So you should have 3 possible boxes you can write in for this question, just to get an idea of who is on the call.
And can we write some free text, Laura, if we feel
Yeah,
like
you can write, yes, you
Yeah,
can write whatever you like. So.
Awesome. Thank you.
GWAS Whisperer, I think I know who wrote that. There's only one. So we have a good complement of data generators by the looks of things, and yeah. Interested bystander, I like it.
That's me.
Wow, you're really leaning into this.
Okay, so it seems like the majority of people here are data generators, which is great because we want to make sure that any standards that we develop are usable, right, by the people who are actually creating the data. So I think I'm going to move on to the next slide, and then the final part from me is just to ask, what's your main hope for the working group for thinking about this meeting, but also in the longer term project.
Lost my window for Menti. What's the icon for Menti?
It's menti.com.
Yeah, no, I'm actually looking at the
Oh,
icon. I
I-
have so many windows open, I can't find it. Oh, wow.
It's got a like blue M kind of looking thing. Or graphs.
So I think what I'm going to do, so these are great things that I think that's definitely an alignment to what our goals are, as Noelle just outlined. And so I'm going to leave this open, and I'm going to hand the presentation over to, I can't see the agenda, so I think it's Maria next.
Yes.
I'll leave this open, so if you have more thoughts about what you're hoping to achieve from the workshop, you'll still be able to add those. But I'm just going to stop sharing my screen.
Hey, thanks. I don't have any presentation. This is gonna be very brief, but I just, as Noelle said, for the last few years of my career, I've been very preoccupied with effective machine lists. We were charged by our funders in 2019 to start implementing these in the knowledge portals, and we started looking at them in the literature. And yeah, what came out was that it is the Wild West, and this really seemed like it was impairing the usefulness of these lists. And so we as a group started finding them and curating them, and over the past year with my colleagues here and at EBI, we put together the first workshop, which I think many of you attended, and that was really great. I'm so happy that we could do that. We also wrote a paper describing the findings that I talked about in the last workshop of all the diversity that we saw in these peg lists. And so I'm very happy to tell you today, so we submitted it to Nature Genetics. We had a fantastic editor and reviewers who gave us some tough but very, very useful comments. And so we revised the paper and it was accepted, and we got the proofs today, so it's coming out very soon. So I'm very happy about that, and I really want to thank all of you, especially my colleagues for all this work, and it's been so much fun working with you all, but also all of you outside this group who are now going to be in the working group and hopefully come up with some community standards. And I just really appreciate all your efforts. I think it's going to be really valuable for the whole community. That's it.
Thank you, Maria. Excellent. All right, I think it's over to you, Aoife. Beautiful, we can see your slides on our end.
Okay, great. Yes, thank you, Maria. It's really lovely to speak after you. So my name is Aoife McMahon. I'm a project lead here at the EBI, and yeah, my team kind of forms a bridge transcontinental bridge between the teams at the GWAS catalog and the knowledge portal. We contribute to both resources. So today I'm just going to recap the workshop that we had back in September. Even if you were there, it was quite a long time ago. So I'm just going to describe a little bit what was presented, what was discussed, pull some themes out, and go into a little bit more detail on the straw man that we've heard mentioned so that we can continue to move forward from that. So forward my slides, which isn't working. There we go. Right, so the workshop was held in the Broad at the EBI and virtually in September. Noelle has already described the aims. I just wanted to note that all of the meeting materials are available. The easiest place I find to kind of access it all is via the workshop notes. So this is the extensive notes that were taken at the time. These are all linked to the slides, the relevant recordings, so you can hop to exactly where you want to go. All the discussions are there, as well as the Zoom chat and everything is there. So I encourage you to have a look at that if you want to get more into the specifics of things. Noelle has already described what the aims were. So I'll skip that. And we are working on writing up the workshop as a proceedings document. We've been a little bit slow about that, but these are the kind of headings that we're working over that kind of describe the various themes that were covered at the workshop. So we had a range of wonderful presenters, many of whom are here today, who described a lot of the landscape analysis of what currently the landscape looks like. As Noelle mentioned, this is a fast-moving space. But the different presenters talked about the range of different approaches that people can take to address this task of going from a variant and figuring out what the effector gene is. We had a lot of people talking about all the different ways the data can be represented, and the diversity in that representation. Noelle mentioned we did a community survey, so the results of that survey were presented. We had some presenters coming from different spaces they were sharing their experiences of how efforts at standardization went in other communities, and we were able to learn a lot from those presentations. We had a range of different speakers talking about what we were trying to get at with these standardization. What are the requirements? What are the needs that we're addressing? And then, we had a proposed straw man standard that we kind of put up there to kind of pick apart and to move towards a better standard. So this is a figure from the perspective that Maria just described. That's been accepted, and it summarizes something that a lot of the different presenters touched on. So it's a nice representation of this, that basically what we're seeing is that there's many different evidence types that people look at when they're trying to figure out what the effector gene is when they're looking at a specific variant. People can use different terms to describe these things. In Costanzo et al., we use the term variant-centric to talk about evidence that is focused on the locus or that starts from the variant. Other people use different terms for this, genomic features, bottom-up, locus-based, but we're talking about the various evidences that come from where the variant is in the genome. So what's the closest gene? What is it in physical contact with? What is the genomic landscape looking like? EQTLs, all those kinds of evidence types. And on the other hand, you can start from the gene and look at the evidence that comes basically from how the gene functions and what it does. And if you think about, I find the easiest way to think about this, it could be completely independent of where the gene lies in the genome. So we call this gene-centric in Costanzo et al., but other people have different terms for this. So these kind of evidences are guilt by association, how the thing is expressed, what it interacts with. Sorry, I just changed my thing. Different evidence that comes from different types of perturbation, be that human disease or mouse models or disease relevance, drugs, and from literature. And all these different evidence types can be aggregated or put together in using a curational approach or a computational approach. That's the basic landscape, and lots of people had different examples and instances of this that they presented in the workshop. A theme that came out again and again, as Noel noted, that there's broad inconsistency in the different evidence types that people are using and how they present that evidence. So what you're looking at here is an analysis of the different PEG lists that Noel mentioned that are in the Knowledge Portal. And a majority of them do integrate different evidence types and present it in a table, but some only present it as a figure or an image, which is has problems with reusability, and some just look at one evidence type. Of the ones that do look at a table, you can see in this upset here that shows you the different features that is presented in the table of evidence that's provided. And again, you see this dramatic inconsistency. So if you think about very useful features of a PEG list, only a small minority of papers actually identify the locus, show all the genes that they considered at that locus, and assign some kind of score. Broadly, it's kind of a mess, again, the Wild West. So this came out of multiple different speakers at the workshop. So we also looked at the results of the community survey, and just want to pull out the kind of things that people are using the lists for. And this is useful to think about. Julie will touch later on the use cases, but people are using the lists as input to bioinformatic pipelines, to generate hypotheses, as one line of evidence in curation workflows, or as a training or validation set for machine learning. So you can see there's a broad range of users and use cases that we really need to keep in mind. We asked people what they valued in different PEG lists. And it's very clear that people value a combination of different things. So a combination of bioinformatics and manual curation and lots of different evidence types. That was the most highly valued type, and above only multiple lines of evidence in either bioinformatics or manual curation. So that's what people value in these lists. Like I said, we had some speakers talking about other efforts. We had Laura Harris from the GWAS catalog talk about their experience developing a summary statistics format. I just want to highlight here that this effort took a long time. This illustrates to us this might be a long-term effort. In the GWAS summary stats world, the GWAS catalog originally accepted them back in 2018. They did a community workshop, much like we are doing, and working groups as follow-ups, but eventually the standard was implemented in 2023. So that's 5 years really from start to finish. Laura emphasized the importance of the FAIR metrics, that the data should be findable, accessible, interoperable, and reusable. And she discussed how these things might apply to PEG lists. For example, should each list have a unique ID? It should be searchable by the study metadata. The metadata should be persistent. We should have a well-defined data model. We learned from a different type of effort. Marina Di Stefano from ClinGen, who's part of the GenCC consortium, told us about their experience in developing a framework to curate gene-disease relationships. And this is in the rare variant space, Mendelian disease. So the first thing she talked about was how they standardized the terminology within the GenCC consortium. This was an iterative approach that required many surveys, piloting, prioritization, refinement, building consensus before final implementation. But standardized terms was key to their process. They then went on to develop a semi-quantitative framework to classify the strength of evidence for the role of genes in disease. And their basic mechanism for this was doing independent dual curation of a pilot set of genes to their proposed framework, and assessing how well they could achieve concordance with disease-specific experts. She emphasized the importance of buy-in. There's no point in developing a standard if nobody uses it. And to do that, they continually engage the community in different societies. And she also emphasized the importance for being able to update things if things change. We also had a range of speakers talking about standardization requirements. And these were basically the use cases, like what are we trying to achieve with this standard? I'm not going to go into great detail with this, Julie will, but we heard different use cases from the perspective of data generators, resources, and different user groups. So I just want to draw out a couple of ideas that came out of the workshop that maybe we didn't have going into the workshop. And one of the big ones that we kind of had to keep coming back to is what is a list? What is a predicted effector gene list? And this idea of whether it's a list or if it's actually an evidence matrix. How to go from a matrix to a list to potentially a gold standard list, and how these might differ for different use cases. This is my attempt at kind of highlighting this for this example locus. So we've got the example here, one locus, one trait. This is our variant we're interested in. Are we actually talking about evidence matrices? So that is the list of the genes with the matrix of all of the different evidence types combined into some kind of score or categorization. Are we considering just one evidence type as a list or a matrix? Or is in fact a list the selected top genes or thresholded genes for each locus? What is a gold standard list? Is it maybe the combination of different effector gene lists to arrive at the one that everyone agrees on? So these were things that came up, and we should continue to refine, I think, what we mean by these different terms. Another idea that came out in the workshop a couple of different times, that we are really dealing with knowledge versus data, and we need to acknowledge that the lists that we're dealing with are subjective. So those are the things that kind of came out of the section of the workshop. Then we went on to present a straw man standard for what the standard could look like for a peg list or matrix. And a straw man proposal, just to clarify, is a draft proposal, and it's intended to generate discussion of its disadvantages and advantages, I guess, and to spur the generation of new and better proposals. So if you want to have a look at the actual files, example files, there you can use this link here. But I'm going to show them, but if you just want to refer to them yourself, they're in this file. So the structure we're proposing for our straw man standard is it's actually a two-part structure, so that we would have a metadata file which describes the actual data file. So in the metadata file, we would identify the source of the GWAS, provide the sources for the different evidence types, and describe the methods used to integrate them. And then in the actual data file, it's a structured table. It includes all the data for all the evidence type. It presents all the evidence. So it presents the evidence for all of the genes, not just the highest genes, and potentially a universal score. That's what we presented on the day. The metadata, again, these are just the things we suggested could be in the metadata file. It's a little bit easier to look at an example. So again, it would have 3 sections. One to describe the GWAS data, the source, ideally using an accession, a trait, ideally mapped to an ontology term. And then for the evidence section, we suggested 11 different categories and other, based on an analysis of the most common data types in the lists that are in the knowledge portal. These are the categories you see here. And then a free text section to describe the method that's used to integrate the lists, integrate the evidence streams. So that is an example of the metadata straw man. For the actual data file, again, it's a little bit easier to look at the actual data. We would have a standard identifier for the gene, some way of identifying the GWAS locus, for example, the sentinel variant, and then the evidence types, which would refer to the metadata file, following the pattern, the category that's specified in the metadata file, and the specifics. So that's how the headers are structured, and then the actual data. And then there's other fields would be available, including ones that would provide gene level evidence, because the metadata file will be a global, it'll apply to the whole data file, as well as a place for the score or the conclusion. So that's the the straw man that we presented. And then we had breakout sessions for people to discuss. And these are the themes, the things that people came back with after discussion. So the biggest one, I think, was a consideration that we need to have some flexibility in these standards over rigid standards. There was broad agreement that a universal scoring system, it would be premature at this point, although useful, but premature. And that the emphasis should be on transparency in the evidence used and how it's documented. There was some support for guidelines over strict reporting standards at this stage. And for example, this guidance could include like how to assign the gene prioritization categories, how to describe methods, things like that. Again, the idea of the list versus matrix was revisited. There was discussion on whether it's best to focus on the full evidence matrices or the distilled strongest evidence peg list. And there was a discussion about versioning, that it's essential to track changes between the new and the updated peg lists, and an acknowledgement that this is very challenging to implement, especially for smaller labs, for example, versus large-scale efforts. So the question about flexibility over rigid standards came up a lot. And it makes sense that, as Noelle really highlighted, this is a rapidly evolving field and there's high variability in what we see. But I think we need to acknowledge that maximizing flexibility can compromise usefulness. So I just want to take a second to reflect on some of the trade-offs that we need to consider in ongoing discussions about what we might want to implement as a standard. So I'm going to show you a triangle now. In an ideal world, that's my slide. In an ideal world, in our recommendations for what a standard would look like, we would be able to deliver all three features, all three corners of this triangle, right? These are important. We want to deliver something that produces high data quality. And by quality, what I actually mean is fairness, right? That the data is findable, accessible, interoperable, and reusable. We want to have a low burden on the data generator. We don't want to make it impossible for people to follow a standard and produce data according to the standard. And ideally, we would also have a low curation cost. We don't want to have massive amount of curation going on to these things. But unfortunately, we live in the real world. And in the real world, if you pick two of these, it by default compromises the third. So just maybe take a second to think about what two you would pick, and then we'll go through and consider the implications of that. So if we choose, we say data quality is super important, fairness of data, and we want a low burden on the data generator. That means the data will probably require a decent amount of curation. And we need to consider, will that curation be possible? So when we're thinking about, will curation be possible, we're thinking about two things. We're thinking about funding. Are we able to fund people to do the curation? Or are we able to incentivize volunteers? Like if we think about the GenCC example, they used expert volunteers. And is this solution scalable? If we want to have high data quality and a low curation cost, that pushes the burden onto your data generator because they need to do more work to provide data according to your format. So then you need to think carefully about how they are incentivized, how the data generator is incentivized to do this work. That might be through their funders, to their journal publishing, things like that. And do they have the skill to do that? And then on the other hand, if you want a low curation cost and a low burden on your data generators, you need to consider that that will in some ways compromise the data quality. So we need to think about what we really need from the data. And this is dictated by the use cases that we will consider. What really do the users and the use cases require? So obviously what we want to strike here is strike a balance between these things. But I encourage you to think about these trade-offs in the subsequent discussions that we're having. So I just want to finish on after we were writing the perspective and considering the discussions in the workshop, we came up with a set of very minimal recommendations that we included in Costanzo et al. And they are here. And we hope these are uncontroversial, but let us know if there's an issue with them. To recognize the importance of lists, we recommended they be included in GWAS publications. These are the minimal things that they should include: gene, trait, some definition of the locus, some quantitative or qualitative measure, and uniform nomenclature, that it's in a single machine readable file. It seems obvious, but it needs to be said. And that we do what we're doing here, form a consortium or working group to work on these standards, and ideally have some place for everything to be available in a fair manner. So I will end there and pass on to our next presenter.
Questions for Aoife or thoughts you want to come to mind before as we transition to Laura?
I have a bunch.
I was just going to say, should we have a few minutes for questions before?
Yeah, let's do that. I think that's worthwhile because I had a couple things. So Eric, do you want to start?
Sure.
Yeah.
On the evidence types, I'm just curious where these fall. I mean, it was pretty comprehensive, but the two that I was thinking of that I didn't quite see there is missense variants or gene burden. That's been one way that people have picked effector genes, that you have a non-coding variant, but it's sitting close to genes with either rare burden tests of coding variants in that gene, or a genome-wide significant coding variant in that gene.
Do you mean in the strawman or represented in that figure that I showed?
Either. There was you had the list of, there was that very nice figure of all the different types of variant-centric and gene-centric, and I do like that nomenclature. But that was one data type I didn't see represented there, so I wasn't sure how you were thinking of that.
Yeah, I can't remember where it fits into in the context of that figure.
Yeah, I can. So that figure was kind of an oversimplification of the categories, but we were including missense. We have one category for in or near the gene, so that would include nearest gene and missense coding variants, things like that. And we did consider gene burden. I can't remember now where we put it.
But I think the best time to discuss those kind of things is when we will go to Laura and she will discuss the different categories that we should include in the straw man. I think that is that's the important place.
Okay.
For them to be to make sure we cover everything, if you know what I mean.
Okay. And what about then? You laid out the question of what the goal is, matrix, list, or gold standard, I guess, and you want to hold that discussion also. Have that hold that discussion on until after the next part. I'm happy whenever.
Yeah. I don't know, Laura, does that fit in your section?
No, we can have that discussion now. So.
Okay.
Right, because it seems like so I think the ultimate goal is to develop these lists so that we can train models, and you need to be able to have a gold standard to train them against. But that seems, at least from where I'm now, that seems like sort of step 3 or 4, and step 0 or 1 is let's get everybody's list so at least we know where they came from and so forth, and then generating the gold standard list seems like the next thing that we would tackle after this. Is that how you're thinking about it, or?
I think that is an approach that we can take a stepwise approach in what we're trying to address. But I think that I think we would like people's input on where they think the value lies.
Yes. Right. I mean, to train models, we have to have lists where people are very confident that they are the right genes. I guess I'm not quite sure how to get there, because I mean, there are ways. So I love the L2G and open targets, because it puts together a lot of things, but nobody's looked through them sort of manually one by one to say, oh, for this particular assignment, it makes sense and it looks real. And I think there's always in these GWAS, there's a subset of loci where the authors say, right, I mean, this makes sense because of this or that, and then here are the other places where the gene doesn't make sense. So I think, yeah, if you're going to train a model, ideally you're starting with the set that is as accurate and confident as possible. But I don't know how we're capturing that unless, right, we're expecting every author to go through every locus and say, right, and at these loci, I have complete confidence these are the right genes. I mean, it's often sort of buried in the text,
Right.
there, but right, not in the table. So I'm not sure how people are thinking about it.
I wonder if that could, where some of the agents and reasoners could actually get the adapters to actually mine that data and pull that back in some sort of structured reporting that that would be almost a secondary thing, like how would you add a layer on top of it that would pull in the auxiliary information, that's exactly what you said, like how they made that decision or how sure they are, that you could then have as like almost a third output, right? You have the metadata, the actual data, well, I guess the list too, and then this sort of like, I don't know what to call it.
I mean, there's a notion you had in there of the manual curation.
Yeah, maybe that's where it is.
Which some authors may do or may not, or may apply to all loci or only some. Maybe it's just enough to allow for that field and capture it.
That could be.
Well, I think Julie will touch on some of the possible things that we could do would be trying to fit existing lists into a straw man, and we can try and see if the different types fit, if we can represent them.
Cool. What else?
Well, I have a bunch, but let me just pick one. And again, I've been really obsessed with the open targets co-localization lately, the co-localization of everything against everything. I'm now convinced that when you assign a gene to a GWAS locus, you have to consider not just that trait, but every trait that co-localizes with it, and that your gene should explain all the traits that co-localize, especially the ones that have a stronger signal than the one you're trying to explain. So I'm curious if you see that fitting into the framework that you've got. It's variant based, it's a variant based thing, but it's branching out in this additional dimension of every GWAS has been done. And previously that would have been really difficult to do, but again, the work of the open targets folks of co-localizing everything against everything makes this possible, and that's a rich layer.
Sounds silly, but could you almost, if you were able to standardize the list, you would then be able to map them all to variants. Then you could run it through open targets. You could actually see what you could use as part of a service that we would do if we actually got the entire community. This is beautiful. Or we could do this with some standards we already have. You could run these lists that we already have, find all the variants, what they co-localize through open targets resource, and then backfill all the current lists with that information. I mean, that would be something that would need to be funded, but that would be a way to do that like operationally, systematically.
Yes.
Am I thinking about this right?
There's two parts to it. One is I guess I'm hoping that in the future, as people publish GWAS, they will do this systematically,
Right.
and that will be an additional sort of line of evidence. But yes, from the existing lists, you would expect that variant-based annotations probably don't change a lot from disease to disease, but gene-based annotations could, because if the model says, I'm trying to explain a cell trait, I might pick this gene, but if I'm trying to pick a psychiatric trait, I might pick a different gene. Whereas if they co-localize, I'd argue that they should land on the same gene. Okay.
I like that. And then the final thing I wanted to add, I really like that triangle. I think that triangle must be up in the corner of the screen as we go through, because I think that's kind of that's the way we should anchor to, because that's what we want to do as this group, kind of balance that trade-off and make this something that is easy to do, but also as accurate as possible. So with that, are we going to Julie next?
Yeah.
Right.
All right. So now I guess we're gonna go off the path a little bit into the Wild West. So I hope you're all ready to come with me here. So we've talked so far about this straw man, right? About moving, developing these sort of minimal recommendations that have been accepted for publication recently. And now we want to move from those minimal recommendations into more fleshed out, formalized standards. So how do we do that, right? We're going to have to work as a working group to evaluate this straw man through in-house assessments, getting feedback from the community, and then work iteratively to refine this existing framework to make it something that's useful for the community. And finally, we're going to have to work together to distribute this to the community and encourage its adoption. So this is what we're thinking for the future of this working group. We'd like to have monthly meetings that are about an hour each. And we're envisioning these as blocks of meetings where we focus around a single topic. So as Eric suggested, we pick something that we think is most relevant to start, work together as a group to resolve that topic, and then move on to the next topic that's of greatest relevance from there. These are some of the topics that have come up based on discussions from our previous workshop and thoughts that we've all had, but we want to hear from you as well. So we've talked about solidifying the data and metadata standards, and that's something that Laura is going to go into during her presentation. Benchmarking and defining use cases, comparing approaches for prioritization, and then finally creating formalized community recommendations. So for benchmarking, this was the idea that Aoife alluded to earlier. This is basically identifying the gaps in the straw man by fitting the existing peg list into it, right? So if we develop, this minimal framework, we take, say, the 149 peg list from the Predicted Effector Genes knowledge portal that Maria worked very hard to curate, and ask, where are the gaps? If we try to, force this to fit into a framework, is it working? Is it not? And do we need to take a step back? A second thing we've talked about doing is defining the use cases and building out the standards according to those use cases. So some of these use cases include individuals who want to submit to a knowledge base, provide input to bioinformatic pipelines, generate hypotheses, confirm independent findings, prioritize gene drug targets. I think a big one for the future is sort of this computational ingest and reuse component, especially for AI, ML, and knowledge graph readiness. And then finally, ways of validating those with AI, and then enabling others to compare the validation approaches. So these are just a few that we have, but we're going to open up the floor at the end of this little spiel so that we can define these out further. And then finally, we want to create community recommendations. So this is going to involve a bit more outreach, right? We need to engage with professional societies to move from, our sort of standards to a set of endorsed recommendations, right? I think we can learn a lot from what the folks at, say, ClinGen have done, with their work that Aoife was talking about, the ACMG AMP recommendations, and other sort of similar community standards to move this forward. We can also work toward a public pilot launch where we coordinate with the community to identify new data sets and apply those standards, so that then once we've developed them, we can showcase them and sort of encourage their adoption. And then finally, I think it's important for us to revisit these periodically, take a step back and think about what we've developed needs to change based on new technologies, new needs of the community, et cetera. So then longer term, we thought about comparing the results that are obtained by different prioritization approaches. And this might be, a bit of a longer haul, so probably not our first step, but the challenge here is the lack of standards, right? So the goal would be to identify data sets, apply and compare the various approaches. This would require positive and negative controls, and a rubric to assess the scoring systems. To do this, we would need volunteers, so data contributors, as well as community members of the working group to test it, with the ultimate goal of defining the gold standard list, as Eric was alluding to. So now I wanted to open this up to all of you. These were some of the discussion topics that we've thought about, but we want to hear what you think. Is this sort of capturing what's on your mind? Do you have other thoughts? You can paste it in the chat or raise your hand and we can open this up. Any thoughts?
Can you go back to the list?
Yeah.
post list. By benchmarking here, do you mean approaches for the framework and metadata, or do you mean figuring out which peg lists are correct and which ones are wrong?
Yeah, so the benchmarking, we were thinking more like retrofitting existing peg lists into our standards to see how well they fit.
Yeah, not evaluating them, right? That's part two, maybe we get excited, we get so lucky. But I think part one is like how do, the retrofitting is a good term. Yeah.
Okay.
Cause I think that could be, I mean, it's an easy activity and straightforward activity that would give us some information on where the gaps are, and so therefore those are things to look forward.
I can't stop sharing so I can see the chat just in
Is
case.
Catalan's kidney scorecard in one of the peg lists?
It is, so actually that's a great idea, so I talked to her about that, actually we saw her in Philly, cause her original list is, but not her new one. Cause she was like, it was too much, here's your use case, it was too much for me to put all of it in one place, so I put it on my lab website because I could spend time and just do it on my own and hold it there. She's like, but if you give me something simple to do, I'd do it. You know, because the last time she worked with us, it was straightforward, and she was like, great, because we were kind of working with her. And she was like, please host it. I don't want to host it. You know, so she's open to that. So she's a great example. I wonder if she could be someone we ask to almost take what did before, what she did now, and how to retrofit it as well into that standard. Because that's a use case as well.
Thinking about that triangle, I mean, she's motivated,
Yeah,
but still, how do we lower the amount of work that she has to do?
exactly.
Yeah.
Exactly. I mean, another silly thought, I don't know how this works because I'm not a computer scientist, but I happen to live with one. How easy, I can imagine much easier for us to actually take lists that exist and to create essentially a reason or a program that would go in and pull them into the format we want them to be pulled into. And would that be an activity that we could do with this group? Like maybe we could do it almost, I know I'm going way off the reservation here, but you could do it as a data challenge where you could suggest to people like, here's all the data that we've curated the peg list, because there's APIs that can be made to all these. You could pull them down and see if you can make adapters that would allow you to put them into some standard format that this group anoints as a way of proof of concept. Am I making sense to people? I don't know if that would be a valuable thing to do, but
I mean, there's a limited universe of lines of evidence, and in theory, a reasoner could say, oh, based on either the column header or some other information that came to the table, map a column to
yes.
one of it.
So I think that may be something worth doing as like a sidebar exercise with this, where if someone's so interested in doing that, could be an activity that we could do.
Well, and then we could even offer it as a tool for the community. If it works well,
Yeah.
then we put it out there and say, you know, do you have these lines of evidence? Do you not want to, you know, spend time developing this out for your own publication? Here's a tool that
How
can
you
do
do
it
it
for
better.
you.
I think not all of the information you'd want in the peg list is always in one table. Often, I know that sometimes like the loci, the coordinates of the loci are in a separate table, and then so when we did some comparisons for this nature perspective, Jason had to write some scripts to collect info from multiple tables and combine it. So, but yeah,
And my other thought was, and this goes back to how much metadata you want to capture. So if I have a column that says mouse evidence,
Mhm.
there are still 100 different ways that could be interpreted.
Yeah.
The primary one being what exact mouse phenotypes am I counting as a hit?
Yep.
And that's not always well documented. So are we content by saying, oh yes, this is a column that links back to mouse data, or do we want to say yes, you have to have those additional layers?
Yeah.
And I guess we have the date that the table was published, because obviously
Yes.
mouse data changes over time.
Right.
So it's the mouse data as of.
That version or that date. Yep.
All right, well, I think we're gonna get into Laura now, but if you have additional thoughts that come up as Laura's presenting, feel free to put those in the chat, feel free to email them after this. We want this to be an evolving discussion with you all. So Laura, go ahead.
Thanks, Julie. So for this part of the presentation, I'm going to talk about how we move from these kind of broad recommendations that we've made to actual standards that we can validate data against. So we want to think about where are the gaps, and I'm going to go into a bit more detail about the straw man proposal that we had, and what I want you guys to do is to think about your sort of initial thoughts about this. So we're not going to come up with a finalized standard today, but we need to know kind of where the gaps are, how we guide future discussion. But there also might be some things that are completely uncontroversial, and we can say, okay, that's great. We don't need to discuss that any further, and we know where to focus our effort. So how with the format of this session is that I am going to do my presentation, then I'm going to switch over to Menti, and I'm going to go back through some of the slides and just let people think about them a bit more and make comments, and then we'll bring those to the floor for discussion, and hopefully that will work. Okay. So yeah, while I do this first part of the presentation, please do think about any comments that you've got to make, and we'll have the opportunity to do that a bit later. So yeah, just a very small recap of some of the key things that Aoife mentioned. So we talked about having these variant-centric and gene-centric types of evidence. And here I'm just going to focus on the variant-centric evidence. So we can talk further on, whether the gene-centric evidence is something that we want to focus on in future discussions. And also just to recap this distinction between the evidence matrix and then the PEG list, which is the top gene for each locus, and then those in themselves can be combined to make a gold standard list. So here I'm mainly going to be talking about the evidence matrix. I should also say I won't be able to see the chat while I'm presenting, so if one of the other moderators can highlight anything that I need to know, just shout, please. So thinking about a variant-centric evidence matrix, as I mentioned. So what are the key components of that we need to define? So firstly, we've got the data content. So for example, this is how somebody might present that data in a paper with the different columns for the different evidence types. Then, of course, we've got the metadata content, which supports the data. And this is typically written out longhand in a publication. And in here, we need to think about what content should be mandatory. Like, are there key features that are really important to include, and how we can make that flexible to include different types of data. Then we can think about the data structure. So things like standard headers and the layout of the table. So for example, having one row per gene or one row per variant. And yeah, just to highlight the importance of standard headers, if that isn't obvious, because even something simple like p-value can be represented in a lot of different ways. And then finally, file formats. So thinking about what format the data should be stored in, whether the metadata and the data should be in separate files, linked together somehow, or should they always be in the same location. So then we had some really high level requirements for the metadata that Aoife touched on earlier. So the metadata must include the GWAS the list was derived from, use standard terminology for evidence types, state the criteria for significance for each evidence type. And a requirement that came up from the workshop in September was whether the list contains de novo wet lab evidence, or is it just a combination of tools, evidence, and data that's been pulled from databases. It should include the details of the method for prioritization, and it's important that individual publications and pipeline generated data should be, they can be flagged in the metadata, but they should be interoperable. So just thinking in a bit more detail about the metadata content. So as Aoife mentioned, we highlighted 3 different categories for metadata. So the GWAS data, which should include the traits, and the things that I've highlighted in bold here are things that I think should be mandatory, but we may want to discuss that. So the trait under study, the GWAS source, which can be a GWAS catalog accession ID if it's publicly available, or more metadata about the GWAS needs to be provided, and the genome build. The method, which can be a free text description of the gene prediction method used. And then the list of evidence, and I'm going to talk about that in a bit more detail on the next slide. So yeah, here again, this is the list of categories of evidence as Aoife described that we identified, actually that UA identified. I should give UA credit because she did a lot of work on this, from the lists that are already available in the portal. And so, for example, we would need to provide the name of the tool that was used. For fine mapping data, we would need to state the LD reference that was used, the software and the confidence interval that was considered, the traits that we used for co-localization, things like that. And this metadata can also be used to indicate when the evidence is de novo or whether it's been accessed from a database. The evidence types could be defined via an ontology. I think that's very much a longer term goal, but by defining evidence types in an ontology, we would it's just really highlighting that we need to define exactly what we mean by each of these evidence types, so that there's consistency between different researchers. So one question that we can address later is whether we're thinking about like how we define a PEG matrix or a PEG list. Do we want any of these specific evidence types to be mandatory? Like, does anyone have a strong opinion that we really need to include EQTL data? And if you haven't used EQTL data, I'm not interested in your list, for example. Or do we want it to be completely flexible to include any kind of evidence sources? And then the second question, should we define a minimum number of evidence sources? So, for example, do we call it an effector gene list if they've only included 1 evidence source or 2 evidence sources, or do we really want to say no? This needs to be a certain number before we trust it. So then moving on to data, so the high-level requirements are that there should be a single table containing all the evidence types, so they shouldn't be scattered in different places, making it difficult to parse. It should be a plain text file, so irrespective of the format of the file, it shouldn't include graphics or any other things that aren't computer readable. The evidence for all genes considered for each locus should be presented. Standard identifiers for genes should be used. The sentinel variant for each locus must be stated. A prioritization score should be included, whether that is the orthozone or a universal score. So, thinking about sentinel variants, we need to consider what the variant identifier should be, and that should also include the GWAS p-value, whether there's anything else, any other information we need to know about the sentinel variant. We should include all genes tested for each variant. So how do we represent those? We've proposed the ensemble gene ID and gene symbol. And another requirement was that the genes should be annotated as to whether they're the nearest gene to the variant or not. In terms of the evidence, that we need standard column headers, and those column headers should refer to the metadata. And then, as I said, in terms of the score, this could be the orthozone or it could be a universal score. So then thinking about the data structure, just some really high level things. There should be one row per variant gene pair. We need to define standard formats for the evidence column headers. And one suggestion that we had is that they could follow a pattern like the evidence type and then the detail. So for example, expression in adipose tissue or fine mapping using PIP. We can think about whether we want to set validation criteria for each evidence type or allow full flexibility. The author's score could be in any format, but obviously a universal score we would need to define. And then just finally touching on the file format, as I mentioned before, it could be a plain text file. The metadata could be in the same file or linked elsewhere. And then we're proposing that the top list of top genes can be presented separately from the evidence matrix. And then thinking about the difference between a matrix and a list, do we want to define criteria for the list as well as the matrix? So what we mean by the PEG list is the summary of results only including the top gene for each variant. So this could be, for example, just the list of variant gene pairs with a tick list of evidence plus the categorization or the score. Or it could have some indication of the strength of the score with the categorization. Or we could, it might be preferable that we only genes include passing a particular threshold are included. So for example, only the strongest genes should be reported, and we don't necessarily expect authors to present every gene if there's insufficient evidence. So then I am going to hop to the Menti presentation and just give me one sec to switch over. Hopefully everyone is still on the Menti from before. And I see there's already some discussion in the chat, which is great. Oh, that's sharing my browser. What is it doing that? Give me one sec. Okay, that's it. So hopefully you can see the Menti code in the chat if you're not already on there. So hopping back to the beginning of the presentation. So we identified there are 4 different parts that we could work on to define a format for a variant-centric evidence matrix. The data content, the metadata content, the data structure, and the file format. And so next, I'm just going to ask you to choose, what do you think the main priority for the working group to discuss around evidence matrices? So that doesn't mean if you strongly agree with one or another, that doesn't mean we won't discuss the other one. It's just to know where we need to start and where the biggest challenges might be. And I do strongly encourage everyone to vote. You can choose. I don't have a strong opinion if you don't have a strong opinion. So data content and metadata content are pretty similar so far. Few people don't have a strong opinion. So I'm not seeing any more responses come in. So I think that is probably where we are. So data content is, yeah, data content followed by metadata content, followed by data structure, followed by file format. And that certainly seems like a logical progression to me. Did anyone want to come in and comment on that? Again, I can't see the Zoom, but Noel, if anyone has their hand up or wants to speak in the room, just shout.
Thank you. It was on mute. Noah.
Adam said, I don't know if anyone saw this, I tend towards not having any single mandated evidence type, but using, multiple evidence types would be a qualifying criteria for peg lists. That's what he was saying. So that's just trying to catch us up. But I'm curious, as people are listening to us, what other people on the Zoom who have not spoken yet, if you want to speak up right now or have any questions, because I have one question about, like, if we were to, between metadata and data content, like, those seem a little fuzzy, which is also why they're probably interestingly overlapping. I just want to, I don't know, how we actually would, I guess I'm more thinking, especially with the remaining time, how do we go from, we sure want to do this, to, like, we got to do this. And I know that's hard. And so that's where I want to make sure we can keep time for that. But so I'm just thinking about that as I'm responding to this. But if folks have ideas
Sure.
on that, I think that would be great for this. Got to.
Okay, well, I can move on because yeah, we've got maybe want to get into the details as well. So then thinking about the metadata content. So yeah, we're talking about we've divided this into the GWAS data, the method, and the evidence types. And so yeah, I've just I put an open question. Does that seem sensible to everybody here? That just thinking about the metadata. So this is like the key information that you need to interpret a peg list or a peg matrix, evidence matrix, or is there anything that we've missed, or does that not make logical sense to you? I think if you think it does make logical sense, feel free to just give me a thumbs up, which you can do on the bottom right hand of the screen.
I keep coming back to the mouse ortholog example where, a researcher may choose to include it or not include it, or animal ortholog information. They may choose to include it or not include it. But if there's a column that says, yes, there's animal ortholog, I want to know how they pick that, what phenotype, what animal, what data source. So
Right.
that's why I put metadata first, because I don't want to mandate that everybody use ortholog information. But if they do, I want to be able to retrace their steps.
Yeah, that's a great point. Yeah, and so that I think that's definitely something we want to achieve here. So yeah, I think so there's a comment here. How about the way of combining the evidence sources? So that to me is the method, like the methods that's been used to prioritize the genes. So ethnicity would be in the GWAS metadata.
Yeah.
So that would be the underlying GWAS that was used. So if you have data in the GWAS catalog, you can link it to a GWAS catalog accession, and that's all that sample metadata and so on is already done for you. But if you haven't shared your GWAS data anywhere, or say it's unpublished data, yes, you would want to specify the sample number, the ancestry, and other case control numbers and other things we need to know about the underlying GWAS. So I think we would get into that in more detail in a future discussion. Okay, so I'm going to move on to the next slide because I think that will come into, so these are the 11 categories that we've identified, and just bearing in mind that we have this other category. So this is intended to be completely flexible for adding any other data types, because I think, and you can correct me if I'm wrong, there was a long tail of the distribution, right? So there were many lists that you looked at that had different evidence types that weren't seen in any others. So it's really important to have that flexibility. So I'm gonna, so first question, have we missed any major evidence types here that should be focused on? As we mentioned, this is based on the data that's already in the knowledge portals, but that of course is a subset of all the data and analyses that are coming out. So PQTL, Mendelian diseases.
And again, on the EQTL, to pick on that, GTEx is one thing, but then to know, is it any tissue or specific tissue or cell type or condition? I mean, obviously there's more and more layers of metadata, and we do want to make it easier for people to annotate. But I guess, is it open-ended enough that even the categories, the depth of the metadata can vary?
Yeah, so I think this is something that we would need to work on in this group, is really going into these evidence sources and thinking, actually, what do we need to include there as minimum metadata?
One thing I want to clarify, actually here we only give example of the EQTL, but actually we think about all the QTL available data. So we don't forget other things, just the example with EQTL.
Oh, okay, cool.
Yeah, EQTL data here, yeah.
Yeah, these are good suggestions.
I wonder if we need to think a bit about some of these categories as well, because some of them seem like methods in some way that you might apply on the sources. So for example, you may have used co-localization on EQTL data. So I might not then be sure, should I be putting this in EQTL and showing is that evidence of how I selected which genes from the EQTL were really relevant because I used co-localization, or is it? So, and I think maybe to some extent the same with TRAS, and I think someone mentioned in a comment like Mendelian randomization. So I wonder if some of these are less sort of sources and more sort of tools that you would use on data sources to, So maybe we might want to think about different categories in that sense.
Yeah, that's a great comment, and I think this also harks back to the necessity to really carefully define, potentially in an ontology. I'm at EBI, we love ontologies, to really define what we mean by each category, and then you have potentially got the scope for having something in more than one.
I guess I interpret co-localization as with not a molecular trait, so a disease or phenotype trait. And yes, EQTL, I guess I wouldn't want it unless it had been co-localized with the GWAS trait.
Yeah. Okay, so this definitely seems like somewhere where we need more discussion, that's a good outcome of this meeting. So I'm going to move on to the next one. And yeah, I think a couple of people have already commented on this, but are there any particular evidence types that should be mandatory to include? And the last category there is none, should be mandatory. So you have that option.
What do you mean by annotation in this one?
Yue, do you want to take that?
Like the deliberate result, or like the variant annotations from any known database.
So variant annotation, that is. So
or PAD or that kind of thing.
Say again?
the results from like VEP or like a CAD or CIF, Polyfen, all that kind of thing.
Right. Okay. So it's kind of separate from the mandatory data category you had about the, saying which variant is the sentinel variant. This is additional information on top of that, basically.
Along the same lines, what about tool?
Yeah, I think that's in there. Yeah, that's a good one.
To like the exist like score generator tools like the POPS or like the index. So which is already the bioinformatician tools other people already developed.
So a bioinformatics method basically is what we're saying there.
In the chat, I put closest gene, but I guess closest gene, like things like VEP, are things that a pipeline should be able to add. If we've got the sentinel variant, then then you can have a script just go over on top of that and assign the VEP scores of the closest gene, and just necessarily have them be mandated.
Hmm. Yeah, so that's interesting, having defined what we mean by a tool, bioinformatics method, that's coming up a bit higher, because I think that was one of the things that Aoife, you mentioned, came out from the survey that we did before the workshop, that the majority of people trusted lists that included both bioinformatics and wet lab evidence. But there's also a good number of votes for none should be mandatory. So that's definitely something we can continue to discuss. Okay, I'm going to move on to the next slide. So yeah, thinking about how we define a PEG list, is there a minimum acceptable number of evidence sources? So the first option is no, it's not necessary. And then you have some other options to choose. And if anyone has a strong feeling about this and they want to chime in, I just rely on the other moderators to flag people on the Zoom.
I'd like to ask if people think specifically if one evidence type is sufficient. Is that something we're considering?
It might not do a very good job, but maybe, one really good evidence source might be better than two poor evidence sources. So I think it's kind of difficult to sort of say, having said that, that's, trying to avoid the question really, which is at some point, I guess, as part of standards around sort of PEG lists, we need to define what we think a PEG list really is. So, and maybe that therefore might need some.
Yeah.
I mean, it might be a minimum of one, but yeah, I think it probably will warrant a workshop discussion on its own.
Yeah, exactly. Okay, I'm going to move on. So then, yeah, this is just an open-ended question. So any other general thoughts about the metadata for the evidence? I think we had a few already around the overlap between categories and whether they were sufficiently well defined. Right, so this comment that the tool, the bioinformatics tool that you're using may amalgamate many of the other evidence types, and I think this is, I don't know, Maria, I don't know if you want to comment on this, because this is something that we discussed before, I think, where it could be, these kind of analyses can be kind of self-consuming in a way.
Self-consuming?
Yeah, so if one method amalgamates lots of other evidence types, and then they in themselves are combined back with those evidence types, and
Oh, yeah, it could lead to redundancy. Yeah, I mean, tools do amalgamate many evidence types, and we did see in some papers that people would use a tool that included, evidence type X, and then they would also independently collect evidence type X manually and combine, use that all to weight the score. So that's clearly not a good thing. So yeah, we need to know which tools go into these analyses and exactly what their sources are.
But it seems like that is something that people do. So maybe that again is something that requires some guidelines.
Yes, we did see that a lot, that people would get the results from a tool like POPS or Depict or something like that, and then use that as one evidence type along with others they collected manually.
Yeah. Okay, I'm going to move on. So I think we are done with evidence. Metadata now. We're not done with evidence. We're done with metadata. I'm just keeping an eye on the time as well. And then thinking about the data content. So these are the things we discussed before. And then I'm just going to, I think we've got time to do this. I'm going to go through each one of these in turn. So any initial thoughts about the variant representation or any other? So we've talked about having just the variant and the p-value. Is that sufficient information about the sentinel variant, or is it important to consider direction of effect or those kind of things? So this is thinking in the actual data table. So I think we said that the genome build would be in the metadata. So these are things that might be different for each variant in the table. So the comment about LD, is the suggestion there that you should include the LD between variants?
That was me. I don't really have a recommendation, but just remembering that so for example, many studies co-localized GWAS variants with EQTLs, for example, and many of them did formal co-localization, but others just said any variant that's within R² of 0.8 of the sentinel is, if that is an EQTL, then we're going to count that. So just something to keep in mind that
Right.
People do look at other variants besides the sentinel.
Yeah, I think that's maybe what I was thinking of with that comment there, whether we want people to include all the variants that they did the prediction on, or all the ones in the GWAS, including ones that they didn't bother doing any prediction on. That seems like it could be maybe a bit redundant.
Yeah, actually, I shouldn't have said all variants, all significant variants, genome-wide significant variants is what I meant.
Yeah, indels are a headache for us in the GWAS as well. Yeah. Okay, I'm going to move on to the next one in interest of time. Yeah, any thoughts about gene representation? So we've proposed the ensemble ID and gene symbol. Just to note that so gene symbols are great, but there are some cases where there are multiple genes that have the same gene symbol, but they've been given different ensemble IDs. So that is sometimes useful, and also there we've included this as Eric mentioned a couple of times, the annotation of whether it's the nearest gene or not. That should be easy for people to extract and see.
But is that, yeah, to me, the nearest gene is just one of the columns, right? I mean, you have.
Yeah, so you, sorry, go on, Eric. I cut you off.
No, just that I imagine you'd have your mouse ortholog column, your EQTL column, and then the closest gene column. So it doesn't seem like it's I mean, it is a gene level annotation, but I don't know that we need to call it out here. It's one of the evidence sources.
True.
Although I guess to Adam's point, if it was tagged onto the gene, if I'm a researcher and I don't like closest gene and I don't want to include that in my analysis, still knowing that that happens to be the closest gene, but it's not one of the evidence sources.
Yes.
Might be an approach. Adam, is that what you were thinking? Or?
Yeah, I guess I was thinking if it wasn't used, if a peg list didn't use nearest gene for some reason as one of its evidence sources, then, maybe it shouldn't be mandatory that defines to get into this set of, verified peg lists. I think most of the time it will. I think it's just a question of whether you force, you have to have that one.
So I think there was a view which might have come from Eric that nearest gene should be mandatory, but I guess it's relatively easy to get that information if you know what the variant is you're talking about.
Yes, I guess if we go back to the full matrix, though, I would be curious what the score the closest gene would have gotten. So if I'm already scoring 3 other genes at the locus and for some reason I left off closest gene, I as a reader would wonder, oh, did closest gene really score poorly against all the blunt evidence? So yeah, so maybe it's mandatory as the list of genes at the locus.
That's a good one. I think that's just the basic. Yeah, I think that's a smart idea.
Okay, I'm going to hop forward to the.
We are jumping over a whole can of worms around closest gene. 99% of the time when I say closest gene, I mean closest protein coding gene. But of course, sometimes the causal gene is not a protein coding gene. But if you always include the closest gene, you end up with all kinds of things that we would never care about. Well, sorry, that 99% of the time we wouldn't care about. We don't have to solve that now, but at some point. Sure. Sorry.
I think that speaks to the metadata you're saying, Eric. It's just important to know how people have done things like what gene list are they considering, how have they, and even, how did they say nearest? Was it transcription start site? Was it gene body? Kind of they've done what they've done, but let's just make sure it's reproducible, and we can then understand, particularly when we come to comparing lists, why are they different? Oh, well, this one excluded this, or this one had and I think the same for the sort of region definition as well. Like we found big differences at some loci if you only include kind of plus or minus 500 kb or a megabase, or what's the definition here. So it's important to make sure we collect those things, I think.
Yeah, 100%.
Yeah. So I think we've talked about the evidence quite a lot, so maybe there isn't too much more to say about this. Just how do people feel about having standard column headers that refer back to the metadata? And should we try and set some validation criteria for each evidence type, or as a minimum require its statement of the possible range of a value? So for example, if the possible range is between 0 and 1, it's a very different interpretation than if the possible range is 0 to 1 million or minus 1 million.
Yeah, I like that, knowing the possible range. I mean, it does feel like, at least for step 0, we're trying not to tell people how to score genes, just let us know how you did it so that we can reproduce the steps.
So Laura, I'm curious, we have 15 minutes left.
Yeah.
What do we have left on Mentee?
So I am
What do you
going
wanna-
to, sure, I am going to skip over the scores. Okay, and I think with the data structure and the file format, we were ranked lower in the priority list. So I don't think we need to talk about that too much now. But I do just want to go back to this idea of the evidence matrix versus the PEG list. And so my question is it important to define a standard for the lists as well as the evidence matrices? So I think it's clear that the evidence matrix is where the real meat of the experiment is, but then at a later point, as a later step in this process, do we also want to define standards for PEG, the lists, or are we happy for authors to just, do whatever nice graphics they want? Of course, they can do that as well. Okay, that seems a very clear vote so far. Okay, good. I know I wanted some things to be uncontroversial. We got one there.
We finished one standard today. Check it out.
Yeah.
That's good.
So then, yep, and then I'm just going to skip over that, and then just to finish up with, having gone through all those things that we've talked about today, is there, we focused on variant-centric evidence matrices and the PEG list that comes out of that. Is there anything that we've missed? Should we be looking at, for example, gene-based evidence? Should we be focusing on the scoring rather than the data structure? Yeah, any other final thoughts that people have got before I finish and hand over to Julie? And you can also, comment in the chat or the live on Zoom.
And we want to keep this up while we go to Julie Love. Can people do it?
Yes, that's an excellent idea. No, I will stop sharing the Zoom, and I believe I'm still sharing the Menti. So yeah, looks like it. Got Yue next to me. I can see what she's
Perfect.
All right, so the next item that we wanted to get everyone's involvement in is planning our next working group meeting. So clearly there are some things that we can unambiguously define, but we'll need a little more discussion on some of these items. So to do this, we will please enter your information into the doodle poll provided here. I think Yue is going to paste that link for me in the chat. So we'd ask everybody to please fill this out. Reminder, this is going to be a regular monthly meeting. So if you can't attend the meeting in June, but are still interested, we'll keep you on the working group distribution list, and you can participate in future meetings as well. Just in summary, we've established the community has a really strong interest in and need for these PEG list and matrix standards. But we need to develop a roadmap in order to enable discovery that can come from them. So there are many different types of purposes and evidence types that are integrated by these PEG lists, and they require input from multidisciplinary experts. So we need a really engaged community of individuals to make this happen. So we want to thank all of you for your interest in this and your passion for continuing this forward. And we're excited to work with you on this. All right. So I think next steps, we are going to record the information from that doodle poll, see what date and time works best for the team, and then we'll get back to you with a summary of today's meeting, the notes that Mackenzie's been taking, and then any follow-up items for next meeting.
Cool. All right. So we have 10 minutes left. I have any thoughts or ideas that come to people's mind along this way that we could do as well. Like I'm thinking of things that if people are interested in actually doing like projects on data we have existing or do some benchmarking on the side, we would love that because I think that would be the right types of proof of concepts or applications that would be helpful for this working group. You don't have to think about it now, but if there's anyone who's eager, or anyone in your labs, intrepid young investigators who would like to do this. Or any just general comments too, like Laura has up in the chat. Let me ask you a different question. We're going to meet in a month, and if you're some of you who are just listening and just really like active to see the community move forward, what would you like to see? Many of us who are here in the room are going to be, working on this, and you all don't have to. You can think about other things or go build your other lists or publish your papers between now and then. We're going to be thinking about this. If we did nothing but one thing between now and the next meeting, what would you feel is the best thing we can do as sort of like the folks who are actually doing the work on the ground that you guys are helping us develop the standards toward? Come on, people, I know you've been silent.
So this might have already been in the previous work, but do you have just a tally of which type, how often each type of evidence is used? Then we sort of work our way down from the most common.
We do. That is in the about to be published Nature Genetics perspective.
Yes. So that's a good one. And then we could do that for the newer stuff that's come out in the last, what, about a year maybe since you did it?
Yeah, I haven't curated any lists in a year.
So that's one thing we could definitely do, or someone could do if they're interested. Because again, we want to make this a publication. We want to make this something that, the community can use. There's like a lot of work to be done here that actually is, publishable and shareable with our community. I like that, Eric. Other ideas?
Well, I was just about to put this in the chat, but I can just say it, that maybe those of you who are listening who have already created PEG lists in the past would like to go back and look at them and see, do they contain some of these basic, types of information that we think are really essential? And if not, would you want to update them perhaps?
I like that, Maria. I'm looking at you, Adam, looking at Lorraine. I know we've worked with several of other,
There's Laz, there's
there's
Adam,
Laz, yep. That's a great idea. I was thinking about that for Catlin too, because of her list.
Mm-hmm.
Anybody interested in volunteering or doing it on the side? Maybe you have already done it as part of your next publication.
Yeah, volunteer.
Oh yeah, thank you, Carl. I love it. That's awesome. Right. You've been nominated. That's great. I think that would be fun. One thing I was thinking of doing is a little bit of extracting what we have in our current knowledge base for the published PEG lists and actually doing some retrofitting. I'm not sure how to do that, but that's a project that I think we could. There's a lot of summer students coming to the Broad in the next couple weeks, so there's some ideas that may come up that would be a really good activity for some of them to do in a 6 to 8 week stint. So yours is
we've talked about sort of low hanging fruit, but I wonder, is there kind of a roadmap? And it kind of sounds like the step 1 or 0 is at least ensuring that we know how the lists are put together. But maybe the next step is then, or maybe this is in the paper, a set of recommended best practices that if you're generating a list, you should include these types of evidence, this type of data. And then the next step after that is, I would think, how do you build a model on top of that or meta-analyze these different gene lists and so forth. But is there an idea of that kind of roadmap of we want to accomplish this and this and this?
Laura, do you want to take that one?
Yeah, so I think that having this input today, I think when we got to the end of the workshop in September, it felt like, there's a lot we could do. And one of the goals of this discussion today is to kind of focus that down and, as I said, identify the gaps. And so I think one of the outcomes from today is that we will build that roadmap. But yeah, I think culminating in benchmarking with existing data sets and this kind of thing. I wondered if it was so I think Ellie is still on the call. Did you want to just comment on how this fits into Open Target's needs and because I think your requirements are more around the gold standards, right? And how we get
Yeah.
to that point.
Yeah, ultimately, I think potentially we create